I just unpaused it.
I can see you laying up. We're gonna get started soon. Yes. Okay. Great. It's my pleasure to David Cuccia, who is, I always forget her official title, Chief Technology Officer, Founder and Chief Technology Officer of Modular. I've known David ever since he was an undergrad, both here working with Bruce Tromberg here at the Wizard Institute. David got his undergraduate degree in physics and then was his graduate work in here at UCI. You probably started you started UCI in 1999, if I remember.
97.
97.
Sorry, here 1999.
1999, yeah. 1999, and so you left there at 2006, right? Something like that. Right? You finished your PhD. So in that 7 years, I think David probably has had experience using almost every technology in this world. So he's done work in temporal frequency domain imaging. He, of course, founded or established spatial frequency domain imaging along with a number of other colleagues here at UCI. He's worked, he's done, I'm sure he's touched with CT instruments, and he's done everything from, developing instrumentation, testing it in, phantom systems, translating them to preclinical animal models and measuring things in humans. His passions are, going from instrument development to fast real-time data processing, developing the electronics, the instrumentation, and the underlying software behind the kind of to give real-time measures of optical properties. He was a dedicated member of our attendee of our virtual photonics meetings, writing in the upstairs conference room, has a real passion for modeling, understanding the fundamentals of light transport underneath it, and developing efficient coding and generalizable coding frameworks to process that data. He is the architect of the initial versions of the virtual tissue simulator. Him and Carol were the first ones to kind of build that initial framework, along with the development of the initial graphical user interfaces that we're using today. So it's my pleasure to introduce David. He's going to tell you about the trajectory of his career since leaving UCI, both the science and the entrepreneurship. So, David, thank you.
Hey, everyone. Thank you, Boston and Bernard, for inviting me to be here. It's a pleasure to do so. And apologies in advance for not being able to connect visually with you and perfectly because there's screens and stuff. But I hope to get a chance to meet everyone here one-on-one tonight. So if you want to stick around, please come and introduce yourself. I didn't totally make it around before the talk got started here. Hopefully Richard can still hear me and moving the mic. So
Yes.
I thought today I was.
David, you don't need to be near the mic where the whole room is.
Oh, the whole room is.
Yeah.
We can hear. It's not this mic at all.
No.
These. Oh, it's even easier. Yeah. Cool. All right. I thought tonight I would break up my talk into a couple different sections and try to just kind of in an expository way show you different parts of what's gone on at my company and how it's translated from the university. It's not really a tech talk on SFDI, spatial frequency domain imaging. I don't know if you've heard much about that.
We'll get that later.
Okay, that's coming. Yeah. Tissue's blurry, we measure how blurry it is and calibrate reflective brightness. And that's the kind of the basis of our measurement technology that's behind what we're doing at the company. But I'm interested in what you guys are interested in. If you had questions about entrepreneurship and startups, or writing business grants like SBIR grants. I'm happy to talk a lot about that because that's how we got started. I've I'm addicted to learning, so I've enjoyed learning as founding CEO and CTO, working with real business people on marketing and sales and finance. I've done a little bit of that stuff. Happy to chat about how that works for us, business models and that kind of thing. Also happy to dive deep on science if you want. So I would just encourage you to interrupt me, and that's your assignment, is interrupt the professor here and let me know what we can talk about that's interesting. But I'll just start talking and let me know what questions you might have. So I have some time. My slides are introductions of myself and the company, challenges that we've run into, and some of the solutions Modulin, my company, has created to try to address those challenges. Modulin is the name now. It used to be called modulated imaging, and so it's the same thing. We just kind of shortened it together so it's easier to type. Tech transfer at UCI has been an interesting and very positive experience for me. I learned how to write patents here, and then I turned around and created a company and licensed them from UCI to start the company. Product development has been really interesting. There's been multiple phases of that. It's ongoing. In fact, on this last Thursday, we had a big product demo for a new prototype that's a handheld version of what we're doing. And so that's always going on. And then I have a couple slides on ethics and engineering and biophotonics I showed last year. I thought I'd just leave them in. It's a really interesting time in science in general, and we all have lots of choices to make as engineers that really steer how technologies are used, and they're becoming more and more in the public sphere that we as engineers, when we make choices on which data we use to train our neural networks, it has real life impacts on populations, and so I care about that, and I'd be interested to chat with you about that. And then opportunities in biomedical engineering, that can mean lots of things. How I see the world is one way, and how I try to adopt new technologies and bring them into our field has been very successful for me. And I'm interested in general what you guys are thinking about and ways that you'd like to be successful, and if there's anything interesting there to talk about. So before I go any further, if we don't connect tonight, or even if we do, please, I'll give you all my slides, but also my contact information. And so hopefully you all will feel very comfortable reaching out anytime for any advice or help or guidance or networking or whatever. So let's put that out there. I'll give you my cell phone and you should reach out. Aman and myself are both graduates of the Beckman Laser Institute. We did our PhDs here. I actually got my undergrad here, as Rawson mentioned as well, Professor Vinagopala, excuse me. And my background's in physics, and I really enjoyed learning about the modeling and computation aspects of light transport, and then being able to be here at the Beckman Laser Institute, where there are so many opportunities to apply that with an animal vivarium and a human clinic, and lots of really interesting faculty that are always coming through. There's always lots of great ideas, and I was just totally motivated by that to stay in grad school, and led to the invention of this technology that I'm still working on 15 years later. Aman has a similar background, but he is way better than me at talking medicine and understanding human problems. And I'm so excited that now he was the first person I hired to come and run our R&D department, so I could do CEO stuff like 10 years ago. And now he is driving, as our CEO, the story forward, talking to investors, running finance models on, return on investment in 5 years, and it's such a cool evolution. But he can talk better doctor to most of the doctors he speaks with, and I'm so excited to be able to keep working with him. Have a lot of great advisors and team members that I can't tell you all about today, but in general, we're very lucky to have an academic background and constant academic interactions with a lot of great collaborators and company advisors. As a company, we're focused on interrogating and making sense of what's going on with the microvascular system. That's a generic term for capillaries and arterioles and venules and the signals they carry. We're learning over and over again that it's really a barometer of tissue and vascular health in any dimension that I've ever thought about it. Even like places that you wouldn't necessarily think to image skin for, like systemic blood volume, hemorrhage loss or something, you can see that immediately the skin is like, first of all, it's very accessible, but it's also super sensitive. You know, what's happening in the skin might have a lot of different signals going on. It might be hard to pick apart what's going on, but at the heart of it, it's a very sensitive organ, and we faint and we flush, and those are our shunts, arteriovenous shunts that are opening and closing, and are creating very dynamic optics that you can see. And it turns out for our diabetes patients that that's actually the root cause of some of the problems our patients are seeing, what we're trying to help are seeing. So, very sensitive organ, great first response kind of organ for targeting. Any kind of dysfunction is usually related to risk of complications. And current standards of like characterizing microvasculature have been very difficult to be quantitative. So they're subjective, non-quantitative, and often time-consuming, human, dependent, training dependent, location dependent. Sometimes it's disparities of care where you have different pigmentations of skin, and some are easier to see what's going on and others aren't. So we're hoping to have some new information on that system that's more objective. One of the really good reasons I think to work on the objectivity is that every 20 seconds, a patient with diabetes has an amputation. It's a super huge medical problem that costs money, it costs lives. And it's something that I think is an unaddressed, medical need. And anyone who has diabetes, probably during their life, they're going to 1 in 3 have a foot ulcer that leads to this condition. And the cost of diabetic foot ulcer and their consequences is greater than 5 of the most costly cancers. So it's like really big problem. Not very many great solutions. A great white whale to slay for the next 10 years of my career. That's my hope. There's 61 million patients that can benefit from any development that we have. So that's one of the reasons I'm so motivated. And, that's a million patients that we think can really directly be addressed with this technology. $80 billion in costs in the US per year that could be addressed, maybe reduced in half someday. Less than 30% of patients, though, get an annual foot check, which is kind of like table stakes, the most basic thing you could do to get ahead of problems that might occur. And that's just because the way healthcare is delivered and the fact that there aren't great, easy point of care technologies to get kind of eyes on problems that are occurring, especially in the lower limb where the circulation is so poor. And on the same token, about 56% is like the latest number, but greater than 50% reduction in amputations is possible with early intervention. Things that have been proven to work, they just are too costly and cumbersome clinically to deploy for everyone. So figuring out who's at risk to getting ulcers, what kind of interventions are useful, that's the name of the game, and that's what we're focusing on. So we've at UCI invented or worked on spatial frequency domain imaging, where we shine pattern light onto skin. We look at how the sine wave patterns reflect, and by the damping of the sine wave, we can measure the frequency-based amplitude damping, and from there we can calculate absorption and scattering properties. And if you can separate those properties, you can be quantitative about the absorption, like you can in a QVET in a chemistry lab. Beer's law, ECL, that L is path length. So if you can measure path length with SFDI, then you can get at absorbance and calculate whatever's absorbing, right? And usually it's oxy and deoxyhemoglobin, lipids, water, melanin. Sometimes it's methemoglobin or other factors. Sometimes it's exogenous agents. So it's a basic capability to be able to take a picture of the distribution of absorption and scattering. Sometimes the scattering is interesting. We're funded on a project for the military to do burn wound diagnostics, where the scattering signal is related directly to collagen denaturation that happens during a burn wound insult. And so right at the point of care, right at the point of injury, you could have a diagnostic that could determine if their wound is so severe that they need surgery, or if it's superficial and they can heal themselves, you can wrap them up and send them on their way. So lots of great things you can do with SFDI in general for absorption and scattering contrast. We're focused on oxygenation and perfusion for patients with potential circulatory compromise. That's what the FDA's cleared our system to do. And in particular, the lower limbs and imaging the feet of patients with diabetes is very useful because sometimes you can see focal spots happening underneath the surface where the hemoglobin content is lower, like it was in this patient. This was an observational study, but 13 weeks later, developed an ulcer in that spot where we imaged before the skin had ruptured, a focal loss of superficial hemoglobin. We're actually measuring hemoglobin in 2 layers with SFDI. That's one of the innovations that's made this feasible at the company. And for a lot of modeling and computation. So we can talk about that more. Another thing you might happen is that the patient comes in, we're reporting like pain in the right leg, and we also see kind of that bilateral asymmetry of oxygen, where one of the feet has much lower oxygen in general or globally. And that might mean that they have a blockage in their artery and there's bad delivery of blood and oxygen to that leg or limb. And there they might be a candidate for a stent procedure to put in something that would open up the artery again. And so if the patient just comes into a clinic and says, my leg hurts, that's kind of there's nothing they can do. But if you can have a quantitative endpoint that says, it's actually because of lower oxygen, meaning there probably is a flow of a source delivery problem, not necessarily a sink problem of poor recirculation, then they can be maybe opted into getting a vascular workup that might cost $300 to $500, something that the healthcare system wouldn't normally pay for. And then you've unblocked that medical pathway. So, those are the things that we're focused on. So here, no visual cues during imaging, but if you see a focal spot, you send them on maybe to a podiatrist. If you see a low oxygenation in the foot, and maybe some high pain on one side, a doctor, not the company, but a doctor could make the decision to send them on to a vascular workup. Question?
So in that figure, there's the focal spot on the ball of the foot, but the oxygen content of the arch seems to be at the same level. So could you explain why one is considered a focal spot and not the other?
Absolutely. I wouldn't say it's not, the other is not of concern. I would say we know a few things about, how we compute the data and where the vasculature can change. So it could be to a normal physiological baseline where you would expect the blood to be lower here than here. Here, definitely, we were used to seeing it much more uniform. One of the things that we were trying to do is build up kind of a database of like normal distribution so that we can have a more quantitative way of comparing the heterogeneity of foot, like how much is normal, how much is not. But it's also possible that area under the arch is also underperfused. And one of the collaborators we're working with is really interested in kind of the local oxygen delivery and the branching points where the vessels break. And it can be different per patient, but there is some general trends of where these perforators deliver blood. And so there may be either with an occlusion measurement or something else, an ability to excite, or identify normal or disease behavior in those zones and map that out. That's one way you can make that difference. But that's a great question. We can also see there's like a hyperperfusion or high HPT in the heel. So making sense of these images on a scale of many thousands of patients is important, and that's something that's going to take us a long time to develop. What we're trying to do is provide as reliable as possible additional insights to a physician that can make a decision on a patient's care, and do it in a way that's as simple as possible that helps them unblock processes. And so we found it's really easy to collect data on lots of patients. My technician going out into the offices, having one physician who's trained on these images with their experience to be able to make kind of disposition of the patients and what to do. And so this is one of many factors that they would use to make that decision. But great point.
Excuse me, question.
Yes, sir.
So you said you talked about collecting data from baseline.
Yeah.
What was the differentiation factor when you were collecting? So how big was your data set for baseline, first of all? And then what different factors were you mapping when you were doing baseline? Were you also doing melanin, different regions, different foot sizes? What was it?
Yeah, good question. So we always measure all of the chromophores. So oxy, deoxy, no, so let's say total hemoglobin in the top layer, total on the bottom, oxygenation, melanins, scattering.
Okay.
And then we, over a range of people, but actually we focused most of our studies in the highest risk populations. So that's one big gap for us right now is that in order to target, fundable studies by a small company, we need to define a high-risk population, and therefore we only have so much data on medium to low-risk patients. It's mostly high-risk. So that's a big gap in where we are in terms of training. But, we generally try to collect as much as we can, and we have good, very low rejection rates. Very few patients aren't interested in participating in studies, but it's early days for us. We deployed our biggest scale of deployment commercially has been down in San Diego, which is south of here, about an hour. And we deployed like 25 clinics and collected, over the sum of measurements, maybe close to like 1,000 patients or something like that. So it's big, from an initial data point perspective, but it's pretty small when we're talking about large populations.
And just follow-up question, what is your target population for this? Yeah,
so that's a good question. It's kind of still being worked out where we can best be impactful. So it kind of depends on the customer. And in our case, there's lots of beneficiaries of a good medical product, right? Patient benefits, the doctor hopefully can be a better doctor. The hospital system can deliver better care. The insurance company can save more money. All of these things. But where we're currently focused is on larger healthcare providers that provide the services that are also insurance companies. And so we're focused there on first on dialysis clinics, because people with kidney disease they also often have diabetes and often at high risk for amputations. And so we're partnering with Fresenius Medical, who has, they've at least invested in our company. We've deployed to their clinics. And that's the population that we've started with. I think in general, we're able to, imagine a world where we're at the primary care, say a place like Kaiser Permanente, they're a group that's the hospital and the doctor network and the insurance company all roll in one. So everybody benefits from prevention, as opposed to some companies and some, physician groups, they get paid to treat patients. So they're not actively seeking prevention technology. It's not their fault. It's just that's the, like the dynamics in the U.S. healthcare system. So we're focused now on those larger companies. But what that leaves is these, individual healthcare groups. And our hope is that as Kaiser proves things out, as the government invests more in prevention, and as Medicare does more to incentivize individual doctor groups to keep to focus on the prevention side through various ways, then more and more people will adopt.
You said you rolled out to a few clinics. How long have you been inside those clinics so far?
Good question. So we had a wave about 3 years ago. We started a wave of deployment over about a year, getting it out, and then the study that we were doing was kind of a pilot study. So the study was actually complete fairly quickly, but that customers retained the devices, and then we then spent the next year after that kind of learning about the data and learning how we could help guide treatments. But I haven't really shown it or talked about it much. Oops. But the other aspect of what I've shown you is that this is our clarified device. It's a cart-based device, weighs about 100 pounds, and it's fairly expensive to build and sell. And so there's only certain customers that would be interested in that performance factor compared to where we're going with a handheld device. So I spent the last year and a half making a miniature version of this. And as we go back out, we think we're going to be able to expand to new. Did I answer your question? No.
Yes, you did. I have a follow-up.
Okay, good.
So over those 3 years, were the doctors able to see like a better retention rate of people going to the annual foot checks or even obtaining new customers?
Great question. I would say, based on our initial assessment of the study, that we had really good adherence to the protocol, first of all. So we were able to very easily get in and out of these clinics and get data on patients over time. Very reliable population because they basically have an appointment with that chair every, Monday, Wednesday, Friday for forever. Unfortunately. And so that was one thing. There's direct metrics on, just customers or hospital systems getting paid for efficient and correct and reliable delivery that we would immediately benefit from. But no, we never were able to fully complete the loop and get feedback on the impact of the device because it's a very heterogeneous network. And so we're working on that problem now. It's like, how do we create partnerships so that when this nephrology group makes a referral to a patient to go to a podiatrist, that we can actually track what happens to that thing. Because right now it's often like a fax order that it's going out. And we don't have a navigator that's following the patient and looking at outcomes. But that's really where we can make a big impact, right? Whether it's a combination of technology and people and a navigation system that can track patients through the healthcare system and find who's benefiting from care, and you can optimize your care so to optimize outcomes for your patients. And so that's something we're thinking a lot about. I think I have another slide just in a second about whole product solution. The solution suite. So we focus not only on the device, but on the viewer software to be able to annotate the images and for the physician, and then think about what recommendations for follow-up care they want, and then analytics kind of service or database that allows us to collect and monitor effectiveness of this deployment on a kind of a population level. And as we gather more information, or as our customers gather more and more data, they'll be able to make smarter and smarter decisions on a population level.
This would be my last question.
Okay.
Does the magnify things like that?
It does, but it's a cheeky answer because it creates PDFs and they can import them. Right now, that's our solution to EHR integration.
Okay.
Our customers every different one has a different EHR, and even though Epic is an important one to support, we decided not to try and create EHR integration for the current product, but we will for the next one. Okay, who's next?
Shashi is next.
Okay, thank you, Shashi.
Yeah, so about the wound. So bacteria generally colonize and they form the biofilm on the surface of the wound. So does SFDI imaging technique can image with biofilm, or do you need to remove the biofilm first and then image?
Yeah, the biofilms, following wounds and being able to address, the amount of bacterial load on a wound and be able to see inside the wound during, the wound healing process is a very complicated process business, and we've essentially decided not to focus the business in that area. We're focused almost entirely on prevention. You could think of it as amputation prevention if someone has an existing wound, but we're really looking at surveillance and being able to identify early circulation loss challenges that could lead to an ulcer, so that you can get in front of the problem. Because by the time someone has an ulcer, they're kind of fixed protocols. There's limited ability to give feedback to those therapies in a way that actually affects outcomes. That said, there are some really cool device companies out there that are measuring fluorescence of bacteria to look at the wounds, obviously measuring the size of the wound and seeing response to therapies. But we focused on imaging mostly intact skin. And if we see an ulcer, it's also a red flag, like why has this patient not been flagged before they got imaged with our device. But we saw that a lot right after the pandemic, that there was a lot of people that had poor access to healthcare, and we were screening or scanning for our study and finding people with open wounds that weren't being treated. So there's a spectrum of things that you can do with optics. we have one business strategy, which is to focus more on the prevention side and partner with these insurance companies that are trying to reduce the incidence of ulcers in the first place.
Yeah, good question. So does it also, does the system is able to find the depth of the wound? How deep is that?
Maybe. I think right now we're looking for topological contrast. The doctor can see if there's maybe a callus, low flow, or low oxygen content, make a decision to send patient to a podiatrist based on that information. We do have one degree of depth sectioning, which is that we measure the hemoglobin in the top layer versus the deeper one, and that lets us see kind of capillary perfusion near the surface versus like large vessel occlusion kinds of effects. That's been a very important contrast element to separate. But we haven't really worked on any sort of depth estimation, except outside of the burn analysis that I mentioned. Yes.
So two things. One of them is, does the shape of the foot affect your measurement? Because it's going to be 3D and
Sure.
SFBI, you're going to have to high calibrate, but like some parts of the foot, depending on where you pick to calibrate, the other parts are going to be kind of bad.
Yeah. Yes, we've published on an approach. Here, Monzar, myself, Sylvain Joux, who was coming a visiting researcher from John Frangioni's lab at Beth Israel Deaconess Medical Center, where we measured phantoms at 5 different heights, and that built up a hyper calibration volume that we could calibrate out of. So we then measure the surface profile of the skin, also with the sine waves. We could phase measure profilometry, and then use the height of the tissue to look up into that phantom data set and calibrate with the virtual phantom at the virtual height that pixel is at. And then for an angle perspective, because we can measure the height, we can also get tissue normals. And so we can calculate the normal of the source to the tissue and tissue to the detector and do kind of simpler geometric Lambertian types of corrections as well. So that's what we do is correct into that kind of virtual calibration space.
I have another question. So what if, you're showing for examples where the hemoglobin goes to 0, but not necessarily that's going to be the case. Do you need a baseline image of that patient? Or can you, if it hits a certain threshold, it just flags it? Because some people could just have lower hemoglobin values to start with.
Absolutely. In general, we don't know. I mean, what we try to do with our documentation and our publications is try to be as objective as we can, and where there are places where thresholds could be set, we want to lean into that. For example, we published with Kaiser Permanente and shown kind of odds ratios of picking thresholds, and found in one case, I think it was if you set the threshold here for this population, everyone to the right would have a 4x higher chance of getting an ulcer. And that was really valuable for that partner, because they want to know when a diabetes patient comes to their endocrinologist at the primary care, or to the specialist, the endocrinologist, if you had an office-based scan that you could do, how could you flag people and opt them into podiatry, advanced podiatric care that they wouldn't have gotten otherwise? And so that was important there. Other times it's probably not practical or useful to have an absolute threshold when you're tracking somebody over time and you're trying to monitor, whether there are changes in the negative direction. You want to catch those. If there's changes in the positive direction due to therapy, you want to be able to see that it's working. And the biggest self-calibrated change is a bilateral asymmetry. So that's one of the key things that our physicians have been keying into when they look at our maps is, if you have very yellow oxygen map over here, but there's a bunch of blue splotches over here, maybe that means there's a systemic delivery problem where there's not enough flow to the tissue. And if it can be correlated with some other clinical symptom like a pain in the leg, then maybe that unblocks that kind of referral. So bilateral comparisons are useful, very useful, with the caveat that not everyone has 2 feet. So some of our patients have had amputations. And so that's a real thing. But thanks for your question, Anna. Yes, sir.
So this is more to clarify. With Clarify, are you making a new protocol? Are you replacing as an alternative for a device that's already exists? for doing topical foot imaging, or is it a completely new system that you're installing in hospitals?
Good question. Is Clarify new, or are we kind of dovetailing into existing things? And it depends on who you ask. If you ask, let's just go through the list. If you ask the US Patent Office, it's definitely new because we deserve a patent. If you ask the FDA, it's just like the other guy because it's safety based on the fact that it's safe and effective, just like this other thing already on the market. That's called 510. If you ask any of our investors, it's definitely new because they're funding us, not the other guys, or not the guys that came for us. Medicare is just like the other guys, because if you're paying them, you should pay us to rotate doctors to use our device. So that's the cynical answer. I mean, a lot of time we're competing with doctors and nurses poking at things and looking at things, and their competition is the visual system, and sometimes that's wonderful, and that's what should be the answer, but sometimes that leads to disparities of care where you can't see circulation problems for people with dark skin, or see a burn underneath pigmented lesions. You can also not be quantitative with them. And in burns and in wounds, more often than not, our eyes can deceive us. There's really fun computational reasons that sometimes this occurs. There's these things called similarity relationships, where if you looked at you do blanket illumination, flood illumination, any color of light, just pick a wavelength, and how much light reflects from 0 to 1. That light might be reflecting 50%, but maybe if it had more absorption, it would be less, but also higher scattering, it would be more. And so there's this fundamental certainty of absorption and scattering. And so we were hoping to decouple those things so you can make more physiologically relevant inferences. The story I just told is not as simple as that, because you can always use multiple colors and do constraint-based computations. But that's one of the things that we're trying to do, is add more quantitation and find areas of medicine where we can really impact a clinical decision. That's the key. Therapeutics are the easiest technologies to get funded and out of the world, because everyone likes something new that can be used to treat better. But the next best thing is to find a diagnostic technology that can be tied to a treatment or a decision. And so that's where we're. What's your question? Yes.
Part of this is also to the students. So in this great slide, I only see one line, which proves that he has a viable company. He has a CPT code.
Yes,
So my question for the CPT code is, did you have to work with an insurance company with it, or it already existed?
Great question. So a CPT code is we've been through this, so it's a code in medicine that's kind of championed through the American Medical Association and all of their boards, and then managed by this group called CMMS, that is, you might say, is Medicare, but that's not technically correct, and I'm not an expert. But it's essentially, Medicare is the US government's insurance company that pays for healthcare for a lot of patients, mostly who are of retirement age and later, but sometimes for other patients with other diseases. And they've agreed to give a doctor $25 every time they legitimately use our device to diagnose potential circulatory compromise, which is our FDA indication for use. And because people have 2 feet, a bilateral scan would be typically $50. The doctor could charge to the government to do a scan. It's based on, it's a PLA code, proprietary lab analysis, which is part of the, it's not the physician fee schedule, it's the lab fee schedule. So it's literally like a lab code. If you go, get a blood test, this is tied to a blood test. Masimo Corporation that makes pulse oximeters, they were the first to get a clearance, or not clearance, but approval. And it's like $6 or $7 for a hemoglobin measurement, non-invasive, but that was tied to an invasive blood draw. The guy that got that done for them, his name was Gary Marston. He came to work for us as our director of marketing and sales, and he came on and helped us to develop this code, which is not just one measurement, but like millions of pixels of that data, and we were able to secure a $25 payment for that. So it existed. We went from what's called a crosswalk, But it was a very important milestone in terms of breaking down barriers to adoption.
You can value his company, right? How many people have died in the how many feet?
Yeah.
Is this PLA code easier than getting a CPT code, or is the process?
It is a CPT code. It's easier probably than if you find a pathway for the lab fee schedule, probably easier than to do it through the proprietary lab analysis than the physician fee schedule, where it's about a physician doing a service, and there are these things called X codes, and sometimes people just will basically get a new code, but it doesn't get paid, and then they have the physician sort of bill under that, and over time you show the use of it and the benefit of it, but that can also be fraught with challenges. So we were lucky to kind of have this, be agreed to, and it means that it can kind of support the business model that we were hoping for, which is kind of a more per use hardware as a service business model, as opposed to just selling capital equipment to hospitals and clinics. Our favorite business model is kind of a subscription service where they get, unlimited use of the device and free and updates to all of the software features and the database access and all that stuff. And it kind of pays for itself through that repayment system.
As for Medicare, does it also work with Medicaid as well, since that's for the lower income, or do you have to go through the different?
That's a great question. I actually don't know the technical answer to that. Most of our, don't be sorry, it's not like I'm being recorded.
Actually, we should take it was established a baseline cost, because if you went to a private hospital, they'll use the same code, but they can actually charge much higher.
Yeah, it's a comp that would be used by any other insurance company going out to private payers. But I don't know, so for someone young that had Medicaid and good question. I mean, often what you see, not for us yet, but as a technology becomes like adopted as a good way to do things, very quickly companies that are like the Blues, that are insurance companies that support both Medicare programs and fee-for-service programs, it's a bad look that they would opt, their Medicare patients into a procedure that their other patients wouldn't benefit from. That might also benefit them if they're not paying for that. So over time, we hope that there's a preponderance of evidence that says it's useful for anyone with certain factors that are risk factors, and we haven't defined those yet. But certainly, patients with diabetes that have had some circulation issues, some other, could be flagged as to be inclusion criteria for scanning. But that's not the company's official position. Figuring things out.
I think we should.
Yeah,
you have a lot more slides.
Yeah,
this is good. It's really good.
No, it's fantastic.
Let's move forward.
SFDI. I guess I do have some slides on SFDI. This is a point to a review paper that we wrote in 2019. We wrote it in 2015. This is Sohachu and Mamazar and myself tried our best to kind of give a survey of some of the major themes that have been developed in SFDI since it's been created. So I recommend you kind of take a look at that if you're interested in seeing all the different directions. But in general, you sine waves or other spatial patterns on tissue. It's really easy to do. Projector, some sort of projector of light. We've literally used a Kodak slide projector, a PowerPoint presentation, all of those things. DMD, digital micromirror devices, the common way people pattern the light. Any color of light, you can do it one color at a time, or hyperspectral imaging, or narrow band imaging, one wavelength at a time. Up to you. And it's really easy to detect with any sort of CCD camera or spectrometer. Replace the camera with a spectrometer, and you have a SFDS system for spectroscopy instead of imaging. And the goal generally is to get absorption and scattering signals separated. But there's lots of cool things you can do with it in combination with other technologies as well. If you do fluorescence in a diffusive sample, you can be more confident in the background optical properties that the fluorophore is emitting in. And so you can probably do a better job of computing fluorescent molecule concentration or quantum yield. If you go with highly temporally coherent light, you can do laser spectral imaging. and SFPI at the same time, they flow and oxygenation and compute metabolism rates. Or you could potentially use the different spatial frequencies of sine waves to depth section and use that for tomographic imaging of the illumination light or the fluorescence light or the coherent spectral light. So there's lots of different directions you can take it. And as you've seen, we've focused at the company on this workflow where we shine light with structured light plus, and that's a couple wavelengths, 1, 2 wavelengths, and then we also use multispectral imaging, 5 to 7 wavelengths across the visible and near infrared, and together combine the signal to do spectroscopy of absorption and scattering everywhere, and process all the data in a couple seconds to get images like you see on the right. The touchscreen interface, Microsoft Surface tablet is how it's on the medical device. And you can interrogate kind of 50 pixel regions to see general averages that light up in these dot circles. And you can see, obviously, spatial contrast, make decisions, and like I said, competitions, color photos. So we always have that co-registered. Under the hood, the data is all in 3D, and we're able to rotate it around. But it's kind of whiz-bang technology that doesn't actually help with diagnosis, so we keep it simple and 2D. In terms of measurement technology, we've kind of added to this the 3 quadrants of measurement when people care about measuring optical properties of absorption and scattering. On the top left, you can imagine putting a point of light in space, a laser pointer on your skin, and in terms of reflected light, you'd see like a cone of light comes back. And that cone shape has to do with how much absorption and diffuse scattering is occurring. And so that's the point spread function of, delta function in, point spread coming out, and you can kind of that's the point spread function. And you can convolve that around if you have a distributed source. In time, you can do a point spread function in time, you do delta function in time, pulse, infinitely narrow, of course, because all instruments are perfect. And then what you get back is this rise and decay at your detector, and you can fit that function to get absorption and scattering separated. That domain, time result, has a frequency domain analog. A lot of that practiced here in Professor O'Sullivan's lab and elsewhere. And there you sweep from gigahertz, from megahertz to maybe gigahertz, and measure the amplitude decrease. And maybe also the phase changes. And so those amplitude and phase together can be used to calculate optical properties. And it turns out these are directly related by the Fourier transform. This might be basics for some people that have had signals and systems. It was definitely new to me when I came into it, but I really liked the signals and system stuff. It kind of made sense. And it's true that you can have a system that measures in one domain, and you can have an algorithm that works in the other domain. And you can kind of mix and match depending on signal characteristics and instrumentation cost and complexity and time, and you can kind of play with that. So a lot of points, time domain systems, in fact, the first thing they would do would be Fourier transform the data and then fit in the frequency domain, because they couldn't really make sense of the high temporal moments of fast changing moments of this function that they could in the frequency domain. So what we did was we said, well, if you can modulate light in time, why not in space? And it was at a time where digital projectors were becoming very common. And so we're able to shine sine waves of different periodicities or spacings and vary that spacing and demodulate that way and get an estimate of the modulation transfer function. So that's the Fourier transform of the point spread function. And that turned out to be pretty useful, very pragmatic ways to get multiplexed imaging in this domain across an entire image with just a simple camera and projector. We can also do that versus wavelength. So here is data collected from a spatially modulated spectroscopy system that we've built that shows, actually this is simulated data, excuse me, but it's emulating data that we've also measured. And if you increase blood parametrically, you can see the kind of contrast you have versus wavelength. And so we add that wavelength contrast along with the spatial frequency contrast. And once you apply your SFDI inversion, you can calculate absorption and scattering. Once you measure absorption, you can fit that absorption spectrum more quantitatively to just linear combinations of absorbing molecules. At the same time, each square here represents an inversion that you might do, and there's a pair. You calculate absorption and scattering. And when you look at scattering, what you see often is a monotonically decreasing signal that comes from the distribution of, you've learned this in your courses and probably know more about this than me many times, but nuclear density, collagen, fiber type shape distribution, cell wall interactions. There's so many mitochondria, can I say that? So many different things that can scatter light, but in bulk, it's often a kind of boring decay, exponential, not exponential, I'm sorry. What's that called? Lambda over lambda not. I guess exponential.
Parallel.
What's that? Power law. Gosh, thank you. I'm losing my words. It's good because I need to go back. So we have a model that models melanin. These are some of the very first multispectral images that we generated with this two-layer model, or three-layer model of epidermis, HPT1, and then HPT2. As it flips to HPT2, you'll see the large vessels. It's a very different structural contrast between those different things. And this turns out to be just enough information to do new kinds of left versus right care delivery decisions. It was very important to segregate the melanin from the HPT1. So we had minimal coupling between hemoglobin and melanin. It's very important to stop HPT1 from HPT2 because they're coming from completely different physiology that relates to different kinds of disease states. And we spent lots of time validating this at various levels. Here's a pig study that we did at UC Irvine where we blocked the artery or the vein. In this case, I think it was the vein, creating what's called venous congestion, where blood flow is reduced by 25, 50, 75, and 100% compared to baseline. This is in the belly of the pig where we're simulating a reconstructive flap, often for patients with breast cancer that then will have a reconstructive surgery on the same day. And so showing sort of a dose response in image form, where you can see with SFDI that there's a very low amount of oxygenated hemoglobin percentage-wise over time, versus with your eye, it's really hard to tell what's going on. Like you can go and see engorged vessels maybe a little bit, and maybe at the 100% you see kind of whitening that happens. But compared to visual inspection, this is much more sensitive and more gradual. And so it's showing you kind of the fact that this is seeing maybe 5 to 10 times deeper than the human eye, and it's differentiating based on spectral contrast, not just spatial contrast. Here's what the Clarify system looks like when it images. Oh, it's not. Oh, there it is. It's flashing some lights. You can see the infrared light that is being used. We're cycling the video. So this is during a cuff occlusion experiment. Over about 10 minutes. And you can see at like minute 3, we block blood flow to the arm, and we're measuring oxygen in the distal part of the arm, or in this case, the foot. And you can see visually the images decrease in oxygen, and every pixel is a number. And so we're tracking that. And over the area of the foot here is the average of all the pixels in the image. You can see that direct response. That's compromised circulation, and as defined by the protocol that we followed and every other device before us that has the same medical indication. And this is the type of data that we submitted to the FDA initially to show that our device works. Here's actually some of the actual data we submitted to FDA. In blue is our device, the occlusion across 18 subjects. When you release, you get kind of a hyperperfusion, hyperemia effect, and then it comes back down to that baseline. And in red is something called TCPO2, which is a voltage measurement device that measures how much blood oxygen is literally breathing out of your skin. So it has to go from the blood to the extracellular matrix and make its way through the skin, through the stratum corneum, and then I can see that. So the dynamics are different, and that's why we don't see exactly the same, dynamics of signal. But shows that it correlates, the two signals correlate in the same kind of circulatory loss. And compared to baseline, we did a box and whiskers plot and showed, at some point here during the inclusion, there was a significant difference across the population of skin types 1 through 6. That we saw a significant change in oxygen. Now, these baseline values, back to that very astute question, are different between us and other instruments that have been released in the same market, and it might be different between different, sub cohorts of patients. And so it's going to be important over time to learn more and more about how those different populations in a variety of disease states present themselves on average and as baselines. This is an example of the discussion we were having about setting thresholds. And so this is that example where if you draw this line, the patients, below this line of HBT1, the superficial hemoglobin, were deemed 4.1 times likelier to get an ulcer compared to not. And so these are patients in pink that did have an ulcer currently, or we're likely to had an ulcer ultimately versus not. Here's kind of a dose response in that same signal HBT1 as a factor of variable. Sorry, my brain. As a function of disease state. So someone without diabetes versus someone that has diabetes versus has diabetes and also presents with sensory neuropathy versus neuropathy and retinopathy. So increasing known clinical risk states. And you can see the HBT1 in the population dropping. But obviously with large spreads in data. And so it's important to keep that in context. But this is the type of kind of validation that we're doing to build up the body of evidence and kind of a community of practitioners that understand the contrast and can help form decision processes and decision support systems that can help impact care. So this is work with USC and also, this is the work with the University of Arizona, but we've also worked with David Armstrong after he left U of A and went to USC, as well as with Fresenius Medical Care that provides dialysis services. They were one of our investors. And at the population level, I'll just get through this because we talked about it, but being able to identify people out of populations is important. Being able to do population analysis is important. And at the upshot, one statistic that really stuck with me in this bigger study that we came back with was that 91% of the people that we flagged with our technology out of the dialysis chair because we thought they were at the highest risk, they wouldn't have had any care otherwise, and they had a 91% correlation to an advanced vascular checkup that would never have been paid for them, confirming that they had a disease that needed to be treated. And in some extreme cases, we're seeing people with very severe illness that should have been caught, but because of just the realities of delivering care and nurses' time and foot checks being so scattershot, we were able to identify those patients. So that was very promising, and we hope that kind of continues to prove out as we get to larger populations. So in terms of, do we have a question? No, I'm sorry. We stopped. Well, I won't stop there because we stopped early. I'll tell you a little bit more about the tech transfer process for the company. We transferred the IP, the patents, in a license form from Beckman to Modula. We developed the technology here when I was a graduate student, formed a company, while I was still a student actually, and raised some SBIR funding to commercialize. We rented space at the clinic here and licensed the technology. The technology came out of what's called a P41 Center, a technology development from NIH. And now we're at 2400 Barranca. You all are welcome to come visit. I'd love to give a lab tour if you're interested. And we've developed facilities there that comply to a quality system called ISO 1345, which means that we kind of trust our process for making devices and that we kind of follow our own procedures. It's validated. We've got 510K and CE marking for the product, Clarify there. But we have ongoing collaborations with UCI and with the Tech Transfer Office as well. Fred Bevilacqua is the most important person in my life early on. He was so inspiring and a postdoc that mentored me, and he and I together kind of did the first SFDI experiments with a lot of guidance from Professor Tony Durkin, who taught us all about the practicalities of commercialization and FDA, and Bruce Tromberg, who was my faculty advisor and mentor. And so he and I were doing some of these experiments early on where we could show by depth sectioning, by shining a variety of spatial frequencies. When you demodulate, you see like the triangle beneath and disappear. We apply some really simple algorithms for sectioning data, which is hard to do in general, but show the contrast of same low, deep versus superficial signals. And that was really fun to work on a patent thing for the core capability of doing frequency domain separation of absorption and scattering. It was great to be here embedded at the university because we could get first class exposure to cutting edge science and medicine. People are always coming through here and giving really fun talks. This was Jeffrey Shuren, who came for a visit. He's the head of the CDRH, the wing of the FDA that's in charge of medical devices. Lots of great animal and human experience and validation, lots of great interactions with wonderful humans like Boston and Jerry Spanier, who mentored me on modeling and computation. And we were able to, through various fabrication contracts and grants, to go from more of a transportable technology to one that was more portable and reliable, that could actually be wheeled into hospitals and clinics. And we could trust not just the instrument, but the software and the algorithms to produce data that we trusted. So we could go out into the world and validate some of our market assumptions about where we wanted to target the technology for commercialization. And so our first product was this Reflect RS device. Some of your labs have one of these, and we're really grateful to your partnership. And we learned a lot about how to make things reliable, and we kind of make a lot of those learnings into our commercial product, our medical product, excuse me. And so we've got these out in the world. Companies like Estee Lauder have measured thousands of people and looking at skin optical properties and light scattering, because appearance is based on light scattering, and so they care about what's going on with collagen. Partnered a lot with Dartmouth College. They've done some tremendous work on fresh tissue biopsy. Wendy Wells, Brian Poe, Sam Streeter now, so they've been a real source of inspiration for us. But everywhere, Notre Dame has a system, and the military, we've done some measurements with them at the the USA Institute for Surgical Research, ISR. And even in Singapore, and some wearable device companies that I can't say. Homolox is actually one of our investing companies. They sell cameras, but they actually invested in us when they were just trying to figure out if they wanted to start some venture investment in optics companies, and now they have a whole venture wing, so that's been very successful.
Excuse me.
Yes, sir.
Why was Unilever involved?
Yeah,
Because of
where are you from? Unilever?
I used to work in Unilever. So
Okay.
Was it because to test the skincare products or something like that?
Yeah, in general, it was. Do you know Manoj Misra? He was the PI in the measurement lab, and he was interested in quantitative ways to measure skin imaging. So they were looked to as a service within the company to do any sort of product testing that was there. So they were trying to build up a cohort of capabilities, and he particularly, what's it called, lip plumper products, where it would create hyperemia, or skin translucency products, variety of things, and so they were just trying to be as quantitative as they could. And also about visual impressions and appearance, and they would like go around the world and scan various populations and get kind of big data sets for people to use for various kinds of A/B analysis. So that's fun. It was actually some of our earliest work on kind of melanin separation was when we were looking at face images. I'll mention, so then I guess, our Reflect RS is the name of the research product. And then we essentially wrapped it in FDA medical software and got our first FDA clearance based on this hardware. So it's called the oximeter. And so the screen was different, but the device under the hood was the same. And we'd already kind of been planning to do this. So we built the whole system under all of these kind of device documentation controls and testing that is important for FDA. And so we were able to get that first clearance that says we can measure circulatory compromise. And then we turned around and raised a round of venture capital to re-engineer, and that's what became the Clarify device. Very grateful in that process to be, able to work with people here at the BLI and elsewhere. So much work has gone into building really good, rigorous measurement systems and procedures and systems of thinking around SFDI. Lots of dissemination and learning, talking to customers by going to trade shows and various medical professions, as well as to Photonics West. We actually exhibited there a couple of years, and that was good. But we haven't been there for a long time. It's been mostly medical focused, and that's just now coming back. We're interested in maybe thinking about the next round of research projects because we've learned a lot about our ability to do accurate measurements and computations with the medical device development. Thinking about the next research project, but sorry, that was a plug. I should retract that comment. Trade shows and conferences. So this is a mom winning a best poster at one of the diabetes foot conferences. And these slides are a couple years old now, but I just wanted to share a couple pictures of people doing cool stuff with next generation things. As we kicked off the project for Clarify, we actually called it the handheld project because that was our goal, to create a handheld device. But given the state of technology, it wasn't like we had all of these great machine vision and AR, VR technologies that we would choose from right now. It ended up being kind of a bread box that sat on a cart. But this was the initial work for Clarify. You see little TFT screens and little microcontroller gadgets from Adafruit, lots of fun prototyping with off-the-shelf technologies to try to get the cost of the technology down. You see here's a slide film with sound waves literally burnt onto them. Lots of late nights making stuff work, designing electronic circuit boards, building algorithms, validating them, iterating. And then once we got to a phase where we were designing the Clarify device in earnest and we picked kind of our stack of technologies, this is the group of engineers and consultants that came and helped put all their heads together in optics, electronics, and packaging and manufacturing to make something that was actually manufacturable and also worked. And one of the working parts is that it was formed to function. Like we did lots of prototype modeling of how people were going to interact with the system, lots of different analyses of like what's the footprint of the average clinic or the average clinician, height, that allows us to kind of pick the dimensions of things and pick software interaction mechanisms. I really enjoyed that area of industrial design that I didn't really ever think about before in a formalized way. So any thoughts or questions about that? At 7:36, we have maybe 10 more minutes or 10, 15?
We've got time. I mean, the bus comes at 8:30. So
30th, I was
so we've got some time.
But I have some
time for discussion afterwards as well. But I think go for it.
Okay.
Yeah.
Do you have a question?
No.
Okay. This is a few slides.
Question.
Oh, great.
Sure. You pay less or more than $500,000 for your industrial design?
Oh, less than $500,000. Yeah, definitely. Fusion Biotech is an engineering services company in Orange. It was founded by our VP of engineering. When we went to the FDA, it took us about 13 months to go back and forth with them on the measurement technology and how it was just the same as everything else, just a more accurate way of doing the same measurement. And in the meantime, we couldn't really afford him. And so he had some other ideas about things he wanted to do. We caught him at the perfect time in his career because he had just sold his engineering service business and was looking for things to do. I called him for advice of like, who should I hire for VP of engineering? He said, well, if you can wait 2 weeks, how about me? So he came on, taught us all about how to like do just the right level of FDA documentation and got the whole package together for our first FDA clearance. And then while we're just sitting here twiddling our thumbs, he went out and created Fusion Biotech. And they just got kind of acquired, but they're still here in Orange and a great group of people. A lot of people we saw that picture was Fusion. And they had a very efficient process. It was very dynamic, interactive. We met with them 4 or 5 times, but in between, they would have done many rounds of kind of thinking and validation, and they teed up like the critical questions or interactions for us, and it was wonderful. In terms of algorithmic bias, I put this in here because it's kind of like the ethics of engineering, and you know, there's so many different ways to talk about the signals we see, and this is not a lecture on that, but this is like a slide event that you can look at afterwards and ask more questions if you're interested, you know, about how you think about your data. I'm interested in how this kind of strikes you. But, you know, there's intrinsic challenges to understanding data. There's, you know, perhaps a challenging computation that you're trying to do. You know, we often do like inverse least squares, nonlinear fits, and you never know if it's converged if you're at a global minimum or you maybe you have limited contrast or sensitivity or no post problem, and you have to do regularization. Obviously, there's data that's noisy, but noisy can mean different things. You know, some people would say, you know, when you reconstruct an OCT image and there's speckle in the image, that's noise. But another person's, you know, one person's noise is another person's signal. You know, just that's actually information that turns out to be useful to measure maps of flow. But you know, it's a fundamental characteristic of your images, and you have to either be able to deal with that or eliminate it or characterize it one way. There's red herring data, like, you know, you might be making incomplete assumptions about what chromophores are beneath the surface. If you have methemoglobin in competing against deoxyhemoglobin, maybe it's just because there's been some hemoglobin breakdown products, and you know, you should have picked 660 instead of 630 nanometers, and you've been less sensitive to it or something. But that's intrinsic stuff. But there's also situational things like algorithms that have been developed with a limited population. So you all know this kind of type of story now with machine learning and everything. You pick. As an engineer, you might have access to certain data, or you might be able to find a study that gets access to certain cohort of patients, or whatever. But then there's always a danger in generalizing, you know, the kinds of things that you come up with. So you know, one way you can kind of just always have as a guideline as a scientist and engineer is to always state your assumptions and speculate out loud. You know, it's hard to be brave about that sometimes, but out loud about the different things that could limit you know, the impact of your claim, but at the end of the day, that's how you build trust, and that's how you do the next thing. So just be thoughtful about it. Lacking of a gold standard is a big one. We've dealt with this our whole, you know, career as a company. Is you can't, you know, we're trying to validate a measurement of, you know, what's going on inside capillaries, and you can't exactly stick a syringe and do a blood draw from a capillary. You might be able to do it from an artery or a vein. Sometimes that's not the whole story. Like if you have blood that skips from the artery to the vein, you could have blood that goes through the capillaries very slowly. And the oxygenation in the capillaries is actually way lower than it is in the vein on average. And so you can make limited assumptions about things if the gold standard isn't really there. So again, state your assumptions, but you know, always be on the lookout for new ways that you can kind of pin down pieces of information and be more and more confident about your measurements. Global thresholds are hard, ranges applied to diverse groups are hard, and again, you can state your assumptions, or you can look towards alternative things that might impact therapies equivalently or better by looking at comparative measurements or trends or other kinds of things like that, or co-registering or co-registering with patient information that's not just your data, but other pieces of information that might help tell more of the whole story. Awesome.
David, I'm working with another group for another skin-related problem using SFDI, and we found that actually, if we want to look at contrast for particular indication, that actually the optimal kind of range or measurement conditions where you see the contrast is actually different based on skin pigmentation. So I'm just curious to see if you use variable thresholds or variable, you know, you're making some sort of conclusion whether this person should be referred to a vascular specialist or not. Is your approach more that you've accounted for the moment and therefore can isolate the relevant hemodynamic parameters, or is it actually the key measurement conditions that give you that contrast are actually dependent or is influenced by the skin pigmentation itself?
That's a great question. I would say, as a blanket statement, as a company, we have to be very careful about guidelines that we put out there. We essentially don't do that.
I see.
We try to work with physicians that can gain experience with the technology and make an informed decision based on their, you know, the totality of their experiences and what they're seeing in the image data, having heard all of our caveats and, you know, documentation on the limited cohorts that we've measured and all of those things. I think that's probably a theme, not just with us, but with others, that, you know, as technologies get out into the world early on, it's hard to make a claim that something would be different in one small population versus another. And so kind of learning from the data at a statistical level is important. And it might be that like different communities are different, and not just because of skin tone or other kinds of age factors or gender factors, but also even just how a specific healthcare system could impact a population. So it might be like, you know, in the southeast, that they have such impacted like podiatric care or something that they need to set thresholds differently just to have like a cost-effective healthcare delivery.
Got it.
It might be that, you know, Kaiser West LA can make a completely different decision based on their healthcare delivery. I'm not trying to make East versus West here. Just, you know, they might make different decisions. So what we're trying to do is create a suite of products that gives insight into the statistics for a population, and then hopefully over time we can kind of interleave. And this is where 60% of your physician cohorts also made a decision to go on left versus right, and start to like put that in. But maybe trying to be really careful.
Thank you.
Sure. I guess the last one was mismatch between the algorithm and use case. I don't know if you'll talk about this in this course, but you probably are pretty aware based on your exposure to various optics-based studies related to the diagnostics that, you know, when you do a study, you can kind of make trades between sensitivity and specificity and how you set threshold, how you might say set clinical decision protocols. And so there might be some diseases or states where, you know what, this is such a high risk, life-threatening thing that you want to tune your algorithm or your thresholds to flag anyone that's possibly has this disease you're looking for. Like melanoma might be an example of that. So what you do is, because you have real world and noise and uncertainty, like you necessarily create more false positives. You're going to flag lots of people, and some of them will have melanoma, and that's great because you caught them. And the others that didn't, well, they had biopsies, they had impact on healthcare delivery because you're investing dollars in people that didn't have the disease. But it was a good decision because it's a high risk disease. Other places, you might benefit from just like picking people out of the population that just have some sort of flag, a completely different end of the spectrum. You know, this very sensitive measurement, and this person is just like off the charts, different than everybody else. We're going to do something off that end to it. program that they wouldn't have otherwise had. And that's kind of where our company is coming from. We're flagging people into more preventative care. We're not really taking any healthcare away. And so that's a different use case. So think about customers, the risks, the types of decisions you're trying to make. Of course, you have to make that fit the real world and how the FDA thinks about risk management, which is this whole thing. If anyone's interested in that for your product or anything else, I'm happy to chat about it, but that's the context. And so here in red are some of the things I see out loud, take homes of ways to deal with that. The other thing I just wanted to quickly chat about is that there's so many different opportunities for careers in biomedical imaging and medical imaging. Obviously, my bent is more entrepreneurship. I've enjoyed starting small companies and working with a variety of people and learning new skills and kind of evolving as we needed. There's others that really enjoy, being a specialist within a large system where they can do their one thing very well, the best in the world. And there's places for everybody. I want to just kind of point to a couple of things that have been hallmarks of my success that I'm grateful for, which is in translating ideas across domains. Like I'm kind of a technophiliac, so I like learning about new capabilities in software or electrical engineering or computer science or optics. And, it's not always the case that people in medicine are looking at those things. And I'm always thinking about ways that we can bring those into our field and make some sort of leapfrog capability that didn't exist before. And so one of the ways I did that was, having insights into projection technology and camera technology, knowing that we could apply some of the same methodologies that we developed in frequency domain photon migration or diffuse optical spectroscopy, where we're doing, frequency domain modulation measurements into the spatial frequency domain. And there we kind of harness all of the same calibration strategies and all of the same sort of, spectroscopy algorithms and Monte Carlo approaches and everything. But it was just, adding in projectors and multiplexing with this newly democratized tech. Projectors weren't new 15, 20 years ago, but they were emerging as really easy and affordable to deploy. The other is that we've really focused at Modulum on developing whole solutions. We talked about Clarify, the device, but it's cloud connected with a HIPAA compliant third-party cloud that we use. We developed a viewer software that a physician can use anywhere to review cases in a centralized place, collecting data from being collected at clinics, many clinics. We've also developed back-end analytics software that we've made available to our customers so that they could see kind of percentage-wise who came in with certain, disease states, what questions did they answer one way or another, and how did that correlate with the quantitative measurements that we made in terms of oxygen and hemoglobin, and make some more system-based assessments, both to optimize their outcomes, but also to optimize their bottom line, make sure they're focusing on the right patients. And tech-enabled digital health solutions. As you go through your career development, you'll see lots of new technologies emerge. And if you're interested in the CTO kind of role that I've had, challenge you to just always be thinking about how those new shifts in technology, how you can anticipate 10, 15 years in the future to something being a commodity that was a really esoteric technology before. And it might not be the case that today it's a commodity. But if you can kind of build a plan of execution that you're either NIH or investors can find value in, and if you can validate those things and de-risk them, you can kind of build today for the plan of that democratization that is definitely coming. It's just a matter of time. And that's things that people like to invest in. They also like to invest in things that will make them money tomorrow. And so you kind of have to figure that out for your use case and your investors. But for us, diabetes is a problem that's just getting worse. healthcare delivery is very disparate and not centralized. We're trying to impact, that by developing kind of commoditized diagnostic equipment, smarts on the algorithm side, to make it so that anyone can deliver this type of healthcare, not just a specialist, and that'll help us scale to patients wherever they are. Because we're trying to go from delivery of that at a specialty clinic to areas, through primary care and retail to the home. More and more will be pushed to the home. More and more will be telemedicine. You'll be developing technologies, I'm sure, that will be used kind of uniquely in the home because that's where people are. And bringing technology to people as opposed to kind of the old school bringing people to technology is going to be so, just kind of a mental exercise to think about for your world, your domain. I would also say that for me, I'm really interested in so many different directions you could take SFDI, and I think underexplored is the increase of contrast enhancement you can get with high frequency information. This is a paper that Aman published as part of his dissertation, along with Silvagio and myself, and it showed how if you put two tubes in a phantom beneath the surface, if they're close enough together and you just shine a plane wave, you see kind of a blob, and if you did a profile, you'd see kind of just this Gaussian-like shape. But if you increase the spatial frequency, you see this, two separate tubes, and with higher contrast and better resolution. And at some point, if you go to a high enough frequency, maybe the signal to noise drops out, and you lose contrast where you have it otherwise. So there's like a optimization problem to do for an instrument here that could maybe tune into objects of different kinds of spatial features to do rapid, surgical guidance. If you had tissue and you were trying to resect something, near a critical nerve or organ, and that nerve or organ had a fluorescence signal, maybe by, dynamically imaging with this, by tuning the spatial frequency, you could get more confidence that if you take away the next millimeter or 2 or 3 of tissue in resection, you're not going to damage that, that key thing that you care about. And so you can go faster. And the business case is, faster surgeries, more accurate surgeries. But there's so many different directions you could take that. And that's enhancing contrast and resolution with SFDI is really interesting to me. And Brian Pogue, originally at Dartmouth, when he give these talks, talk about contrast detail analysis and how, for absorption and scattering kind of differently, and they have different contrast at different spatial scales depending on the baseline optical properties. And so you can kind of design your instrument and your algorithms to optimize for the problem we're trying to solve. Others that I'm working on now explicitly are burn diagnostics. I'm grateful for collaboration with Vanderbilt University that generated the data on the left, which showed the burn index primarily related to optical scattering measured by SFDI, correlated well with histologically verified burn depth by Menton staining to look at the crosshatch pattern of the collagen or whether it was intact or not. That work has been kind of evolved on and worked on in earnest at BLI here under Tony Durkin's group, and they applied machine learning algorithms to the SFDI data set and showed the ability, even within like a single burn wound that's heterogeneous, to be able to reconstruct the different areas of full thickness versus partial thickness versus superficial versus unburned, creating these pseudo-color maps on day one that really correlate pretty well with, ultimate wound outcome. That shows how you have a diagnostic technology on the point of injury that could make a decision that was correlated with ultimate outcome a month later. And so we've been funded at the company by the military to work on a burn wound diagnostic system and primarily focused on miniaturization. So this is going to be planned to be like a far forward field deployable, for military units that are embedded in places where they may not be able to, get their troops out very quickly. And so being able to triage large number of civilians very quickly and decide, who can be best helped first is very important. And finally, just thanks to all the people here and that have helped the company in the past, and all of our employees here at various holiday parties and celebrations. Tell stories about each of these people that I won't. Here's our current lean team. It was really fun. Kevin Seeger's our electrical engineer. Raj is our data scientist extraordinaire. Also really great with imaging and optics. Kyle, our VP of engineering, who's all about mechanical systems and quality systems and runs the medical device building side of things. And fearless leader Mom Nazar. So thanks a lot. I have cards here that I'm happy to hand out. Please, let's chat. Any questions for David?
Right. So I think it's really inspiring, first of all, great talk. Great to see that I think it's always difficult to hear stories on the sad side of research that has, they've gone out but couldn't work out, but you're still there. And
Still,
I was want to know, when you started off after your PhD, when you were working on, I know there's a lot of things to do, especially coming out of research and spinning into a company. While you were going through FDA and you were making new prototypes and you were trying to get investors and for you doing a bunch of more things.
Sure.
What was the premium focus that you were putting on, and what was the thing you would do differently if you go back, just to optimize the whole situation and get it correct?
Great question. Thank you for the question. And I mean this in the most positive way. I'll share with you that a 12-year-old just interviewed me and asked exactly the same question. So we have some really promising biomedical engineers coming up here now in the near future. I'm sorry, I don't mean that. That's a great question, and it's almost the hardest one to answer because how do you? I haven't done statistics on my life. It's just one, n equals one. I would say, every 6 months or so, the company's focus shifted a little bit. And if there was anything that I've learned, first, in a positive way, is like a good signal to keep track of in terms of like iteration is keep track of the rate that you're learning, whatever that is. Like it changes all the time, right? But if you find yourself 6 months later doing the same thing and you haven't learned the next thing yet, that's probably an indication that you should be changing a mindset or pausing and not wasting too many resources until you answer the question it's been nagging you for the last 6 months. Not always easy to do. We have some multi-year questions that we're unable to answer without a big partner, and we're looking to try to drive that. But that's one thing. I would say that the answer on what I would do differently, I think I was really happy to follow the kind of modern startup and entrepreneurship world as I was growing up. And one guy, Steve Blank, is known well in the community. He was subsequently, based on his success and whatever, hired by NSF to create this I-Core program. And his core thing, no pun intended, is this business model canvas. And you can just find it on Wikipedia, but it's just like literally writing down on one piece of paper all the different aspects of your business. I annually knew about all of these things. They were going to be important, but I didn't force myself fast enough, I think, to really be self-critical and also spend resources kind of proportionally across all of those different concerns. It's like, who are your customers? But that's not just like doctor. It's like really segment who's going to buy it, who are the early adopters, who are the laggards, how what you're doing is going to deliver value specifically to them. Why are they going to give you cash? Who are they giving cash to now? And what should they give their cash to you instead? Because it's usually a zero sum often. Not too many risk takers out there, but Yeah, I'll just leave it there. It's a business model canvas, and really thinking about all the aspects, the whole product solution, and really validating and focusing on learning in each of those areas. And you can kind of go through your learning metric and say, well, that's the same as it was. Let's figure out if we were right about that, or figure out the right order of operations for whatever you're doing. Hopefully that's good. So we went slower than we could have. We probably burned, it's being reported, but tens of percent of government funds that could have been expended, more focused toward the solution. But I'm pretty proud of what we've established with government funding. We got almost to our first FDA clearance with just government funding, and that put us in a really great position for venture funding, where we didn't have to, answer basic science questions or basic technology questions when we were going out asking people to take risks on a business model.
Was it follow-up?
Yeah, it was
Follow-up, you should.
Okay, so follow-up is just a bit more specific, something that will definitely not ask. How was your first approach towards an FDA 510K? What are the things that were really essential for you to keep in mind at the end, and what are your takebacks with your first meeting, maybe a Cuccia meeting that you had?
Oh, good question. Yeah, so Cuccia was a way to talk to the FDA before you submit your 510K. Yeah. We actually made a kind of a strategy decision based on the fact that there was a very long history of people getting devices cleared with our specific claim language, that we thought it was sort of rolling the dice and just kind of kicking the can down the road to have a Cuccia, where maybe we were overconfident, but we were confident that if we prepared the package and submitted, then if there was a problem, they would kick it back and we would have the conversation then, versus have a Cuccia where you submit time and money, you dedicate time and money into this process ahead of time, and then learn from the discussion with them, and then kind of change and pivot. There's some time there where you're twiddling your thumbs, and time, it means salary for the company, and so it can be an expensive decision to make. And so I think it's a good decision for us. It took us a while to come to terms on the fact that we had SFDI being added as a technology underlying it, but at the end of the day, it's producing a metric of tissue oxygenation like our predicate device did, and it was like a superset of what our predicate device had. So we ultimately came to a kind of a good agreement on that. But that's how it went. We've tried to, most of the time we're doing like strategy stuff like that, just take one risk at a time. So like, you saw that we took the research hardware and got a clearance for the software. That was risky. We weren't going to go to market with the research system, but what we were able to do is like pin down the FDA risk so that we could go to investors and say, okay, now we've got FDA clearance, now we need to build a device. And then the device that we made, Clarify, actually took us only 29 days to get cleared, as opposed to 13 months for the previous process. We went through a process called a special 510K, where you can compare it to your own internal predicate device. And if you can compare on industry accepted conformance standards, as opposed to have to do clinical measurements and stuff like that, then you can get a very rapid approval, and that's what we were successful in doing.
While you were doing the FDA clearing, were you looking into other products to diversify your business into, or were you just kind of more laser focused into one?
Good question. We entertained a conversation here and there about the technology capability that we might want to build into our device, but we've had way more trouble trying to decide what not to do within our own technology and device. We've got this, what we think is a measurement platform with a variety of potential applications. And one of the challenges early on was figuring out order of operations and which of those we were going to focus on. And it's possible we even spread ourselves too thin and could have focused faster. It's part of that business model canvas conversation. We knew from the beginning that our first product wasn't going to be our last product, that we were selling research hardware to faculty members that wanted to practice SFDI. Kind of call it a postdoc in a box, and that you could learn as much as you could about SFDI, and maybe in a year or two have a cutting edge system, or you could buy a system for us and get started with the hypothesis driven research that you care about for your field. So that was our first product, but we knew we were going to a medical product the whole time. That said, there's lots of spin out, potential spin out technologies or simpler or different technologies that we've envisioned. And it's been hard but important to focus and not work on those things because, we're 15 years later, we're still trying to solve that very first kind of success story. We have a lot of internal successes, but being able to show that you can round trip a large population of diabetes patients with a large customer as an insurance company and a hospital provider, takes a lot of motivation. And so that's our big, North Star. But in the meantime, we use research hardware all the time, and we're interested, as I said, in disseminating that where it can make sense and people are actually interested. And maybe there will be other kinds of spin outs or startups based on the core technology. We just need to find some space and time to do that. Does that answer your question?
Yes, it is.
Question.
How accurate can you measure melanin given that the spectral extinction coefficient is just indicate there's no spectral features to it?
Yeah, melanin's hard, and we are trying, every day to be better and better at it. I think we'd say we have something that's highly competitive or better than most that we've seen. But it's something that is, like you say, it's nondescript. It's monotonic decreasing absorption. Melanin and phenomelanin are slightly different. It's not just a scatterer necessarily. Sorry, it's not just an absorber necessarily. You have melanosomes that act as scattering reflectors. So you see kind of a real bright signal from melanosomes reflecting back, which is kind of counterintuitive. So I think it's an important problem to work on, especially for the disparities of healthcare and giving accurate information for our machine learning overlords to make good decisions. You have to have good quality data to start with, and so we're really focused on it, and we hope to continue to update algorithms as they get better within the implementing stuff. And luckily, they're starting to change how they are thinking about it, creating new mechanisms to allow you to do what they call post-market changes. Kind of what they call good lab practices or good, manufacturing practices. This concept of documenting how you go about developing new things. It's possible to get a clearance within certain bounds that says, once you get data from this population, you're allowed to update it within this kind of machine learning protocol that you've kind of outlined for yourself. So we're interested in that. That's possibly a way to update. Can I ask you in return? Do you have thoughts about this?
You definitely see the impacts in data, but like you said, melanin is monotonically decreasing, so it's kind of difficult to recover, especially because then the part of the spectrum that is going to be the most important is in the beginning of the spectrum with deoxyhemoglobin, and we're trying to sub-modify them at a time. We have hemoglobin problems, but we definitely are thinking really hard about it, especially for the future as we explore the number of comforts that we recover.
Totally, and depending on what device it is, it's going to have different sensitivities and different ways to address the problem. A contact probe versus a planar imaging system could have very different implications.
Yeah, but we definitely see the effects of melanin. You can see in oxygen tissue saturation if even if you just flip your arm.
Sure. Oh yeah.
In the back of the arm versus the front of the arm, same person.
Absolutely.
Thank you for your talk. I have a simple question about technical. So when I see that experiment data and I can as you explained, they have a little effect based on the distance, like from the detector and surface of the sample. So from your data, I can see the middle of the probe already they have about the others. Yes. So I just wonder the other surface was the same, but how about if we use if we ask the patients standing on the device like a scale, and then take a picture from the bottom.
Sure.
Then the surface all flat, some space part may be a little bit different, but most of surface it should be same plane.
Yeah.
They might be give more better result because or most of the surface is uniform, and also it might be easy to combine with other like for example scale and then in-body device. So have you ever thought about the way to make it?
Yeah, no, it's a good suggestion. It's something we thought about. I think it comes down to what you're trying to measure and how you're going to measure that most accurately. And for us, we care a lot about the HBT1s, the perfusion in the top layer of skin, and how that's related to how these arterial venous shunts might be depriving the local tissue of oxygen and oxygen-rich blood. And in that case, if you stood on a scale, most likely you'll just push away all of the blood and our signal goes away. So we're not comfortable designing scales and contact systems, but we've definitely thought about it. And for other types of measurements, it might be exactly the right thing. For example, if you're trying to get, I don't know if you've seen a demo of it, but we've got in vivo clinical multi-photon imaging system, very high resolution. Have you guys seen that yet?
Yes.
And the MPM system, and there, having a contact kind of technology that registers everything makes it possible to get really good high resolution registered MPM images. So it might also be true, like that, to have a dynamic measurement with and without pressure, either to look at capillary refill or to look at the skin with and without, the blood volume. Maybe there's a way to, look at the ultrastructure of collagen in and around a tumor in a new way by looking with and without blood contrast. Maybe that's a new contrast mechanism. So, that's what I would say in general. It kind of depends on what you're trying to measure. For us, it's been a challenge to think about how it would work. Our main concern is that, when a patient comes into the clinic, they're acclimated and stable enough to make a measurement that we trust. Someone's freezing and they're cold, their feet are white because they're just cold. That's different than actually having very low total hemoglobin as a baseline. So acclimation protocols and being in a clinic for a certain amount of time, protocol of taking a patient's shoes off in the same time that you're within a certain time window and that kind of thing. It's all very important. Maybe simple or mundane, but important for the diagnosis. Thank you.
I still have more questions.
Me too. No worries.
I will run. We'll have to join issues. Okay. So I think more on both of the areas, there are always going to be some technical limitations, whatever you develop. I think as a former CEO, when you started working on tech development and then you went on to actually build your business model around it, how were you deciding when to stop technical enhancements and then make the business around it, or maybe focus to reshape the business or pivot to something else, or maybe just enhancing the technical expertise to the point where you are the best in the market and there's no one to beat you? What was your...
That's a great question, and I would say that's always evolving for me, and I've never been comfortable. I've either been too worried that we're engineers just learning engineering and not stopping and moving on to the business development. I'm worried that we've stopped working on engineering when we could be making leapfrog kind of progress and make the product 10 times better if we were just... And so I've learned both lessons, and there's no right answer. I think kind of gauging how your business is learning is the general guidance I'd go back to. It's really easy as technologists, so love doing technology. And if you're an entrepreneur that's trying to create a business, it's something different. And you'll have to be sad and put down technology to build a business and sometimes. Or maybe you find the perfect partner to either run the business for you or to take over for the science and engineering for you. So a lot of it is about building relationships of trust and competence. We've often, met people at business conferences, and based on good, positive conversations, develop relationships and maybe engaged in consulting, and then they finally came on as employees because they really liked what they were doing with us, and we really liked what they were doing. We have a chance to dig build those things, but those things that take time and trust, and that's hard sometimes too. I've seen lots of businesses fail because the CEO wanted to do it all themselves and he wasn't really willing to share or be reasonable about equity or all those things that can corrupt relationships. So, yeah, no good answer. That's the summary.
All right. For my last question, so what would you say would be some key takeaways from your experience that you would give to like new or just starting?
Key takeaways and advice. Well, I'd say the best piece of advice I ever got, I don't know if you have this neurosis that I shared, but like I was really worried early in my career and I was making the wrong decisions about what to do next. My chemistry teacher, I said, come see me because I did well on a test. And I told her, like, I like science and people. And in high school, I thought I should be a doctor. But then I saw my first surgery and blood and guts were bad. And so I was, in a crisis about that. And she said, you need to meet this guy, Bruce Stromberg. And Bruce introduced me to all of these new kinds of ways that you can impact medicine by building new technologies. And it's very inspiring. And so he kind of helped me make that decision about what to do next by exposing me to new things. So that's part A, is just make sure that you're always, excited to learn something and inquisitive about something new. Even if it's not your main thing, if it piques your interest, take some time out of your day or your life to dig into it, because you'll probably learn something. Even if you don't do it, you'll learn something about what's valuable. But the other part is just like not be afraid to make a mistake. I told my advisor, longtime family friend, mentor, he took me to lunch when I was trying to decide if I wanted to go into grad school or get some experience with private industry by doing an internship. And he said, David, you can't make a mistake. And I thought he was telling me, like, don't F this up, like, not make the wrong decision. What he was saying is the opposite. It's like, it doesn't matter what choice you make, you're going to make a decision, and it's the right decision because you're going to learn from that experience. And the only thing to do is to make sure you're brave enough as you collect new data and that new experience to change directions. So a medical resident I was working with here in a research protocol, and she's like, I really hate being a doctor, but it's too late. That really stuck with me. It's like, it's never too late to change. You just have to kind of be brave enough to make that change. There's so many different ways you can be impactful in the world through policy or science or engineering or creative arts. Like, don't be afraid to make mistakes, to learn lessons and make new choices. That's the generic guidance I give you. Cool. 3 for 3. Well, thank you guys so much. I got my cards here.
We got cut off today.
Okay.
When you make friends with the